Okay, we're back. So in this section, we're going to go a little bit more into the chromosomes. So the first section, I gave you kind of a molecular overview, the DNA, what's happening with the DNA, how you replicate the DNA. When we got into the second part, I started talking more about mutations and what changes, how you change the DNA. And in this part, we're actually going to talk about something called cytogenetics. And I'm not going to go into great depth and detail on it, but cytogenetics is basically just kind of a branch of genetics that's concerned with studying the structure and the function of the cell at the chromosome level. So what's happening to the chromosomes that are affecting what's going on in the cell, right? So first, let's use a couple terms. When you see something called a karyotype, by the way, a karyotype, this is an example of it. Here you can see all the chromosomes. So you can see all the double pairs, they're color-coded so they match. It's 46 chromosomes altogether. You can see that when they do this and they look at this, they call it a karyotype. And in this karyotype, they've actually color-coded it. And you can see some of the banding. So you can see some of the dark stripes, light stripes, dark stripes that are kind of codes. But it's really cool when they do this. So of course these aren't this color inside of your cell. They pull them out and then they code them or color them on their own. All right, so the karyotype is just a spread of all the chromosomes. Usually you take one cell, you extract it. Usually it's a lymph cell. They pull it out and they catch it in that phase called mitosis. So mitosis is, remember, all the chromosomes are condensed. They're lined up across the center of the cell, and then they can split that cell open, split the nucleus open, and just rearrange them, let them all shuffle out there. And that's called a karyotype. When they look at each of these cells, kind of like this, they'll refer to it as the P and the Q branches. So you've got this P arm and the Q arm, or branch. They call the P the petite because it's shorter. If you measured here to here, it's actually shorter than from here to here. So here's the centromere, right in the center. So going from the centromere out, that's the P arm. Going from the centromere out, this is the Q arm. P is for petite, Q is for the other. There's no actual name for the Q. Right? At the very tips, we talked about these before, the tips are wound up with these things called telomeres. And the telomere, remember, is just that cap on the end of it that kind of protects it. So again, here's the real picture, and then here's just an example. And you can see X and the Y chromosomes. These are called the sex chromosomes. X and then Y. Of course, women have two of this X. A man has a Y and an X. So in this situation, who is this taken from? It's taken from a man. Okay, so mapping the chromosome. Now that they've pulled it out, they've done their karyotype, they can actually take and look at all of the genes in it. They can color code and they can see bands or striping. So when they color code it, they see darker areas, lighter areas. And when they do that, instead of color coding it all pretty like this, they actually just color code it like this. So you can see one genetic area is here, another gene area is here, another gene area is here, and then these are all sections of genes. Here's our centromere. When they number these genes, they number them from the centromere going outwards. So what would this be if it's a short arm? It would be the P arm. And then this would be the Q. When we identify where that gene's at, we call it the locus. The location is the way to think of it. So the locus is identifying the location. So if we give this chromosome a name, we'll say the X chromosome. So here's half the X chromosome. Remember, you have another exact match of it over here. But here's the X chromosome. If I wanted to go and say that where we know the genes for Duchenne, Becker muscular dystrophy, funny that I picked that one because that was related to a question that we already answered in the last section, or in the first section, muscular dystrophies. So here you have Duchenne's. We know the gene for Duchenne sits right here. So we would say that it was at the 21st locus of the X chromosome. That's how they label that. Okay, so that's the location. Alleles means that these genes aren't all exactly the same. Alleles are telling us they're different types of one specific gene. So you may have a good healthy gene that makes proteins in a muscle here, but you may have a dysfunctional allele, which means that same gene is there to make proteins in a muscle, but it's different than the normal one, and it's called an allele. It's kind of like when we talk about different types of hemoglobin, or we talk about hair color. Hair color is actually a perfect example, because with hair color, you can have brown hair, you can have blonde hair. You have genes that will code more for the proteins and pigments in the hair. Okay? They're different alleles. If we're going back to the recipe example, an allele would be like if a gene is referring to the cookies, the allele is the type of cookie. Is it chocolate chip? Is it a white chocolate chip? Is it a butterscotch chip? So those are alleles. Right. And then this just shows another example. So here you have all the same gene, but when you look at, see how they're all wound the same length, similar codes, what they're actually coding for is something slightly different. So if this is for making color or pigment in a flower, You have one allele causing the red coloring, one allele causing the white coloring, one allele causing yellow coloring. Right? So putting the big picture together, remember, the DNA is your whole book collection. The chromosome's like pulling one book out of here, and we'll say it's a cookbook. The gene is the recipe section, like the dessert section. We'll just say this is the desserts, right? And then the allele are the types of cookies in that dessert section. So that's breaking it down for an analogy. So we'll keep using the term allele over and over again. That's why I'm trying to point it out now. Okay, so as we're going on with the genetics, you want to know the terms genotype and phenotype. Genotype refers to what your genetic code is. What does the recipe say, word for word? Does it say, 2 eggs? Does it say 3 eggs? Does it say 2 cups of sugar? Does it say whatever? That's your specific code. That's what your genotype is. However, what you actually come out with is the phenotype. So genotype is what genes you have, but the phenotype is what you can actually see. When you look at someone, they may have genotype that says they could have brown or blue eyes, but when you look at their eyes and they're brown, that's their phenotype. So that means that they carry the genes for blue eyes, right? So if you want, this is kind of a fun game I found. You can type this in or click on it, and it's a little game that helps illustrate genotypes and phenotypes. It's made for kids, but it's kind of fun just the same, right? Another terms you need to get familiar with are dominant and recessive. So dominant means that if that allele is dominant, that's the one that will show. That's the one that leads to a phenotype. Recessive means that if their genotype is recessive, it's hidden. If there's a dominant present with the recessive, the dominant dominates, and that's what you see. If there are only recessive genes, that's what you see. So I'll show you a little bit more later. But dominant means that, like chocolate chip cookies are kind of the dominant cookie because everybody seems to like chocolate chip cookies, where white chocolate chip macadamia nut are recessive. Not so many people have it. But if you were in a store and you had to choose, you saw the chocolate chip and the white chocolate chip, let's say the chocolate chip smells delicious, it looks delicious, it's the dominant one, that's the one you go with. When do you take the one you don't like? When that's the only choice, right? So recessive, when recessive genes are the only option, that's when they show. That's when they become a phenotype. Right? There's one other option, it's called codominant. So it's like having the best of both worlds. Codominant means that if you have a tough time deciding between the chocolate chip dominant and the white chocolate chip, which is also kind of dominant, you can actually make a cookie that's both white and dark chocolate. A good example of codominant is blood type. So let's say your mom gives you type A and your dad gives you type B for your genes. Your genotype is A, B. Your phenotype will actually be A and B because neither of them are more dominant. O is considered recessive. So when you have an A from mom and an O from dad, your blood type is actually type A blood because that's what you see. When we look at your red blood cells, we see you have type A. But your genes, your child could actually have type O blood because you could pass down that one gene. So dominant means what is more powerful, the one that's going to show if it's present. Recessive means it only shows if there's nothing dominant present. And then codominant means they both show at the same time. Okay. 2 more terms, homozygous and heterozygous. Homozygous means that the pair are exactly the same. So here's a chromosome. Where you look at section A, on the other arm of this chromosome, section A will be exactly the same gene. That doesn't mean that they're coding for the same kind of cookie. Remember, you could have 2 alleles. If this is coding for cookie, this is coding for cookie, this might be chocolate chip, this might be white chocolate chip. Here's where your dominant comes in. Which one is going to be the phenotype? Which one will you see? You'd see the chocolate chip version, right? So homozygous means they're both exactly the same. That means this one is exactly the same. If this is white chocolate chip, that has to be white chocolate chip. Heterozygous means they're different. If this is chocolate chip, this can be white chocolate chip. So hopefully that makes sense. So when you look at recessive genes, the recessive genes only show if you're homozygous for that recessive gene, like type O, the blood type on the last page. If mom gives you an O and dad gives you an O, your blood type's going to be O. Because both those genes are recessive, but that's the only gene you have, so you're going to take it. If mom or dad gives you an O and the other parent gives you an A, you're going to have an A because the A is dominant. If they give you a B, you're going to have the B because the B is dominant. If mom gives you an A, dad gives you a B, you get AB, right? So A and B, it's considered heterozygous, and the phenotype is AB blood. Right. So the next part, Mendelian genetics. So we've gone from molecular to the chromosome, and now to Mendel. And Gregor Mendel was an Austrian monk, and he, in his free time, he studied bees and plants. He loved it. He loved gardening. He loved beekeeping. He found that when he was trying to study bees, he was really curious on how they reproduced and what traits they would carry on. But bees were way too complex. So he looked more at plants, and he looked at these pea plants, and he found that pea plants typically have white, pink, or red flowers on them. And so he looked at those traits, like the color of the flower, and then he would grow them. Actually, I think it was snapdragons when he did the flowers, and peas when he did the pods. But when he would grow them, he'd look at what traits they passed on to their offspring. And he started coming up with this little way that you could predict the odds of having a certain color offspring, right? So Mendel, actually, when he was doing these research experiments by himself, of course, he was a monk. He was more into religion than science, but he had a curious mind. He found that there are two major principles. There's one that he called the principle of segregation, and that means that homologous genes separate during reproduction. So if you have that chromosome and the two sides are homologous, they separate. Right? The other one was principle of individual assortment, which meant that you randomly shuffle those genes, and you never know where they're going to go. So you can take two parent plants and have offspring that look very different from each other because of this random shuffling. You couldn't predict which one was going to be which, but you could predict the percentage. So I'll show you this example with human situations. We're going to actually play a little bit with something called a Punnett square here. So Punnett squares are nice because they actually let us use Mendelian genetics, or things that are observable traits. So when we look at this, there are a few rules. You want to remember the dominant and recessive. When you write a dominant, you write it with a capital letter. When you write the recessive, you write it with a lowercase letter. So in this situation, would this bunny be homozygous or heterozygous? It's homozygous. What's it have? Does it have the recessive or the dominants? It has the dominants. So it has two dominants. It's going to have this dominant trait. Okay? Here, this bunny has two whats? Two recessives. It's going to have the recessive trait. What if this bunny had a capital B and a little b? What's it going to have? It would have the dominant trait. Okay? But its genotype would be carrying recessive genes. Right? So we're going to use this example. When you do a Punnett square, you have to look at the parents, and you can predict the offsprings. So if I take this B, and we'll say that this is for big ears, because that thing has really big ears. And this is for littler ears, so a little smaller ears, but fatter apparently. Right? So this big-eared bunny here has BB, that's the dominant trait. This is the small but fat-eared bunny. When she passes on one of her genes, remember meiosis is going to split this, passes one of her genes on to her offspring, there it is. When he passes one of his genes on, there it is. So now this baby bunny is going to have the big B and the little b. What size ears does it have? Does it have the big ears or the thin, fat ears? It has the dominant big ears, right? The dominant capital letter. Same thing here. Mom passes on to this baby. Dad passes on to this baby. What kind of ears does this baby have? Big ears. It has the dominant trait. It has the big ears. Every one of these, 100%, are going to have the big ears. 100% of these are what? Homozygous or heterozygous? They're heterozygous. So every one of them carry the trait for the thin or the small but fat ears. So they're carriers. We'll talk about these a little bit more. So take a piece of paper. We're going to write this down. I'm going to walk you through how to do a Punnett square briefly. So you're going to draw a Punnett square, assuming brown eyes are capital B, dominant, which is more complex than this, and blue eyes are little b, which are recessive. Okay? When we describe the genotypes, we're looking at the genes that these offspring carry. So let's say that this parent here has a dominant brown and a dominant brown. What color are their eyes? They're stuck with brown. They don't have any options. They only have brown. This one has a capital brown and a little b, which is blue. What color eyes do they have? They have brown. They have the brown dominant color, but they carry the trait for blue. So their phenotype is going to be brown. Their phenotype is going to be brown. But let's see what happens to their possible children, right? So when meiosis happens, it splits these chromosomes in half, right? So this chromosome with the capital B is going to go to this kid. This chromosome with the capital B from dad is going to go to this kid. So this kid has capital B, capital B. That's its genotype. What's its phenotype? Brown eyes. This kid's going to give it a capital B from mom, a lowercase b, blue eyes from dad. So it has a capital B and a lowercase b. What's it have? it has brown eyes, right? So it got brown eyes from mom. It has brown eyes, but it's carrying that gene. This one was homozygous because it was two capital B's. This one's heterozygous. This one has brown eyes, but it carries the trait for blue. How about this one? This one gets a capital B from mom, capital B from dad. It's determined to have brown eyes. It's homozygous, brown eyes. Its phenotype is brown eyes. So now we have 3 kids with brown eyes. These 2 are homozygous. This one's heterozygous. Right? How about this last one? Capital B from mom, lowercase b from dad. This kid has brown eyes, but it's heterozygous. It carries a gene for blue. So all of these kids have brown eyes, but 2 of them are heterozygous, which means their kids can have blue eyes. Right? You can do this in reverse. You can actually predict the parent's genotype if you know the phenotype. Right? So if you predict the parent's genotype, let's say that a baby's phenotype is blue eyes. Mom has blue eyes and dad has brown eyes. So baby has blue eyes. What can you assume right now about baby's eyes? Baby has a little b and a little b. Remember, the little b is recessive, the blue eyes are recessive. So for it to have blue eyes, it has to have the little b genes. So we know this is a little b, right? So we can automatically assume a little b from mom and a little b from dad. So these are both lowercase b's. But it also says dad has brown eyes. So if dad has the blue gene here, what's he have to have here? He has to have the capital B brown gene. We know dad is heterozygous with brown and blue genes. Right? It says mom has blue eyes. Yep, so dad has brown eyes, mom has blue. We already know mom has a little b here. For her to have blue eyes, what does this have to be? It has to be a little b. She's homozygous. So now this baby has little b, it has blue eyes. Okay? This baby, since it was a capital B here and a little b here, it's heterozygous, but it has brown eyes as a phenotype. Since this is a little b, this is a little b, this baby would have blue eyes, right? Homozygous blue eyes. Since this is a capital B and this is a little b, this is heterozygous, but the kid has brown eyes. So it's interesting, by knowing this one kid here, you could actually plug in what the parents' genotypes are and then figure out the rest of the kids. All right. Let's talk about diseases and how that applies to diseases. So carriers, I use that term. Right? So a carrier is somebody that has a disease gene, but phenotypically they're normal. They look just fine. So they have the gene for that disease, like in Tay-Sachs, there are 250 or 1 in 250 people running around there that's a carrier, but that doesn't mean they have Tay-Sachs, right? They're a carrier, right? So for a person to demonstrate a recessive disease, they actually aren't just carriers. They have to have the 2 recessive bad genes. Sickle cell is a perfect example. Because with sickle cell, you can get one gene that leads to the sickle cell. It's recessive. But as long as you have a good, healthy gene from one of your parents, you don't have sickle cell disease. Right? And sickle cell disease, by the way, is sickle cell anemia. It talks about the shape of the red blood cells going from nice round to get this sickle shape, and they stick together and they clump together. Right? What's kind of interesting about sickle cell is that if you just have one trait, which means you're heterozygous for the sickle cell trait, means that you don't get malaria. Right? And I'll talk about that in the next slide. So let's figure this out. We'll say that here's a nice healthy gene, here's a healthy gene, here's the disease gene, here's the disease gene. So both parents are carriers. Right? Neither of them have the disease. But if you look, when they have offspring, 25% of their offspring will have a capital A. Perfectly healthy, that offspring is no longer a carrier. They have healthy hemoglobin, and they're not going to carry on beyond that point. This kid's going to have one healthy gene, but one bad one. Since this is recessive, they don't have the disease, but they are a carrier. This kid has one bad gene and one good gene. They don't have the disease, but they are a carrier. So now we have one kid that doesn't have the disease and doesn't carry it. We have 2 kids that don't have the disease, but they are both carriers. And the third one, they have the bad gene from one parent, bad gene from another parent. They are homozygous, recessive for the disease. They have the disease, they're carrying it. If they were to have children, their child is definitely going to be a carrier. 100% sure it's going to carry. Right, so that's how you use the Punnett square to try and figure these things out. Oops, I forgot to put these in for you so you can actually figure out which of them are. So if we were breaking down percentages, what percentage of these kids are normal, healthy, non-carriers? 25%, just this one. How many of these kids are carriers exclusively? They're healthy, but they're carriers. 50%, so 2 out of 4. How many of these kids carry and also have the disease? 25%. So 2 heterozygous parents have a 25% of a child being normal without carrying that recessive gene. They have 25% of that kid being homozygous for the recessive gene, and then they're 50% guaranteed to be heterozygous. So like I said before, malaria carriers have something called sickle cell trait. Sorry, malaria carriers. If you have sickle cell trait, you can't get malaria. There's something that changed about the hemoglobin that makes it so that the malaria parasite doesn't want to get into it. So you get malaria by a mosquito bite. It gets through your blood, it goes to your liver, and it starts growing eggs and developing those eggs, basically baby parasites in there. Those baby parasites get out into your blood and they go into a red blood cell. They start growing, developing, and they burst your red blood cell. Malaria does that to you. There's something about having heterozygous for sickle cell, you're called sickle cell trait, that makes it so that this red blood cell is inhospitable to this parasite. So the parasite can't get in your red blood cells and can't damage it. So basically, evolution has made you resistant to malaria if you have sickle cell trait. Unfortunately, if you have sickle cell disease, then your red blood cells will stick together, burst, pop. And here you can see the malaria parasites inside of a red blood cell and how they're destroying it. Right. And then one more before a question. So red-green color blindness is another example. What's interesting is that, so red-green color blindness, it codes for red and green photoreceptor pigments. And there are different varying degrees of red-green color blindness. But it's carried on the X chromosome, just on the X. So the red-green color blindness is on the X chromosome. There are lots of different color blindness out there, like 20 different types. But for this one specific type, it's carried on the X. So the Y chromosome doesn't have it. Right? So as long as the child has one non-affected X, their vision's normal. They see colors just fine. But if both of their X's are defective, then it's a problem. Think about this with a male. How many X chromosomes does a male have? It only has 1. So if it has this affected X, all it takes is 1 gene, and presto, that kid has the disease. But with girls, can girls get color blindness? They sure can, but what has to happen? Both X chromosomes have to be affected. If one X has the disease on it, or disorder on it, and the other one's healthy, the one that's healthy actually overpowers the bad one. So what I'm going to have you do is I'm going to have you take this disorder, and I'm going to have you plug it in to Punnett square. So you're going to draw a Punnett square, assuming normal color vision is X, marked with an X, so like this, and it's dominant. Where color blindness is X with a D for disorder after it. So here's a disorder. So you can see mom here, she carries it, but she doesn't have it because she has a healthy X. So dad doesn't have the disorder. You're going to predict what is happening to each of the kids. So what is this kid? What's its genotype? And then does it have the disorder or not? So is normal color or not? And you're going to do that. And you're going to predict the genotype and the phenotype. And then what you're going to do is you're going to see if you can predict the parents' genotypes if, so in this one here, a female baby phenotype is colorblind. So this is a female girl. You know that the female girl is going to be XX. So you may as well write the XX in here. And if she has color blindness, what do you know about those X's? Are neither affected? Is only one affected? Or are both affected? And then you can predict the parents, right? And we're going to predict that neither of the parents are, actually, does it say? Yeah, we're going to predict that the dad is colorblind, but the mom is not colorblind. So baby's colorblind, baby girl. Dad's colorblind, mom is not colorblind. Forgot to type that in. All right, next. So complex inheritance. This is a slide that's kind of an overview, and it's not, I don't expect you to remember every detail and every disease associated with it, but I want you to remember the terminology. So far we've actually been talking about single genes, right? Complex inheritance is not single gene. It's not one gene. It's actually several genes. And how genes interact, if you remember your physiology, they interact a lot like hormones do. So I'm going to describe this a little bit. So first, multifactorial means many factors. There are lots of factors that can affect genes. It's not just the gene. It can be the environment. It can be different genes. So how genes interact. And here are some subcategories of multifactorial. You have something called polygenetic. So polygenetic means many genes. In other words, some features about you require lots of genes to work appropriately. For instance, your hair. Your hair is not one gene, it's actually 4 genes. They have to work together appropriately to give you hair color. Skin color is the same way. There's more than one gene there. You can have multiple alleles. So multiple alleles can affect what's going on. Remember A, B, and O? You have an A that's affecting you, the B that's affecting you, that can be codominant. You have the O that can be recessive compared to these two. So it's more than one. If you have the gene for A and the gene for B, you have AB. Right? Remember, if you have A and O, you have type O phenotype. You can have things that are called complementary genes, where genes depend on each other. They work because the other ones let them. If you remember with hormones, we call that permissive. One has to be present for the other one to work. That's multifactorial. Right? There's collaborative genes, where 2 can actually interact. One does one thing, another one does another thing, but when you put them together, they actually have this synergistic effect where they do something completely new, something different. So when we talk about hormones, it was called synergy. Here they work together. Okay? You can have something called epistasis, where one gene will actually shut off or compete to turn off another gene. So like I said before, some of these characteristics, you already have the concept down from hormones, just apply it to genes. Some genes compete. They shut each other off. Some genes work well together. They synergize. Some genes have to be present for other genes to work. So they're permissive. Okay? And then multiple alleles means different types of genes. And then polygenic means many genes. Okay? And then another thing that affects or makes it more complex is mutations and epigenetics. And mutations, we've already talked about a little bit, it's when you change the genetic code. When you're looking at epigenetics, your genetic code is intact. You may have perfect, healthy genes, and we'll talk about this later, but something happens where you turn off a gene, or you don't allow a gene to work properly, or you turn up the function of a gene, and we'll talk about that just a little bit. And it's kind of a new study or new research area. Another big picture to remember when you're talking about this single gene versus multifactorial, single gene trait like disorders are typically more rare. You might see something like 1 in 10,000. Like cystic fibrosis is a single gene disorder. We'll talk about it later. And it affects about 1 in 10,000 people. Where multifactorial disease could be something like heart disease. How many does that affect? 1 in 10,000? No, more like 1 in 3 people get heart disease. So heart disease is actually a multifactorial disease that has a lot of things going on. So there's kind of a big picture. Single genes, more rare. Multifactorial disorders, very common. Oops. Sorry, that was a slide that shouldn't have been there. Okay, so now we're going to talk about pedigrees. So when you look at multifactorial disorders, the Punnett square doesn't work anymore because there are too many factors. I mean, you could do it, some multifactorials, people will use a Punnett square, but it's just too complex. So it's unpredictable, really. Pedigrees give us an overall picture. It's not always the predictability like a certain absolute positive, but it kind of gives us the idea that, hey, this is a common disorder. So when you look at pedigrees, there are only a few things you want to remember for now. If you really get into genetics, you can go back and look later. But so first, when you're looking at these shapes, it looks like a weird code or a weird drawing, like hieroglyphics. The first thing you want to do is look at the sex. If it's square, it's male. If it's round, it's female. I always think of square being male because I was told once that all men are blockheads. So there's your square, right? And then female, you have those nice delicate round curves, right? So there are some characteristics to remember. The second thing you want to look at is the shading. So there are different ways that people shade. Sometimes they'll shade like half of it or the whole thing, and it means different things. So shading means that person has the disease, right? So they have whatever that disorder is. So some people, like I said, over here you can do half shading and it represents autosomal, heterozygous, recessive, which means that half their genes are good, half of them are disease. So in general, when it's dark, that means that they're an affected individual. Right? When you have this branch in between two on a horizontal plane, it means they're married, or they're a couple, or they are the ones that made it. I guess I just put marriage in there, but if they made it and they produced offspring, then they're connected by a line. Okay? Then you go down. These are each of their offspring, so they're children. Here you can see a girl, a girl, a girl, a boy, a boy. Whoa, look at that. What happened here? It means that boy married someone. It's not connected, so their family tree is not connected in that way. It's only by marriage. When you look at this disease, the very first or the as far back as you can trace, the very first person with the disease is called the proband. So this proband's who passed it on. And when you look, it's got 1, 2, 3, 4, 5, 6 kids. 50% of the kids were affected. Right? Next thing you look at is, did it affect just women? No. It affected 2 of their female children, but it also affected a male. Right? So you look at information like that, right? And you just keep following it down. So you're following the heredity of that gene. You can keep passing it down generation to generation. Right? And then you can see lots more marriages, lots more grandchildren. Here you can see that none of the kids are affected. It doesn't mean that they're not carriers, but they're not affected. So we can take a pedigree and we can actually apply it to a single gene disorder that you could use. For a single gene disorder, you could use a Punnett square, but you can also use a pedigree, so it's just a little bit more advanced. And this Huntington's disease, remember that triplet repeat that happens over and over again? Here's actually the Huntington's DNA. What's really nasty about Huntington's is every generation gets it worse than the one before. So that DNA keeps repeating. You have a child that has it, their repeats even longer. And we'll talk about this in the nervous system a little bit more in detail. But going through here, in this example, they shaded everything, but you can see affected versus unaffected. So reds means they had it. So who got it? Dad or mom? In this situation, dad got it. And then Huntington's, because it's a single gene disorder, with single gene disorders, typically about 50% of the children are going to be affected by it. So, and I'll go over that in better detail later. But here you can see there's 1 child, 2 child, 3 child, 4 child, 5 child. So about half of those, 2.5 kids. So it's either going to be 2 or 3 typically, based on statistics. So here you've got 1 affected, 2 affected, 3 affected kids. And then this affected child got married. Look, one of their kids was affected, their daughter. What's interesting about Huntington's is that Huntington's doesn't actually show up until about 35, 44, somewhere in that range. So midlife, basically, it shows up. So at that time, you've already had children. So you never know if the kids are going to have it until they hit midlife, or they can have a genetic test for it, which we'll talk about later in the nervous system. Right. You can do this with things like breast cancer, heart disease, which is kind of cool. They do it a lot with heart disease. Is that now we're getting more specific. Since we know some genes that contribute, you can take something like heart disease. You can look at, by the way, the slash means they died. So you can look at the carriers, you can look at the people that had the disease, and now we can actually add the genes to this pedigree. Right? So this is just an example where they put the genes on. And then you can actually make this pedigree rounded, and you can see it in an overview. So people do a lot of things with pedigree to make it look more interesting or make it more easily accessible for them. You can look at breast and ovarian cancer, so you can see how they spread. Right? And you can see breast cancer. These people had breast cancer. Right? These people had ovarian cancer. These people were carriers for it. So you can look back and actually identify some of the carriers based on your family tree. So you can see here this person had breast cancer and passed it on. Well, this one didn't get cancer, but he was a carrier. Okay? She was a carrier. She got cancer, breast cancer. She got ovarian cancer. So pedigrees are handy, and I'm not going to make you do a complex test with pedigrees, but just understanding the general idea of pedigrees. So if I asked you a question like who is the proband, was it male or female, you could go back and look at the very first one. In the situation, who's the proband? Mom or dad? Mom was. She was the first one that had the disease, and then she passed it on generation to generation. And this is actually revolving around the BRCA gene. Right? Polygenetic traits actually demonstrate, they can be quantified. Missing a word here. Demonstrate quantification, so they can quantify it. When I was talking about hair and skin color, as an example. So father is African American, mom's pale Caucasian. Okay? There are many genes that go into their kids. That's why the kids don't look specifically like mom in skin color or specifically like dad in skin color. They're actually a blend. It's because skin color has this scale where it's not absolutely dark skin, absolutely pale skin. There's a lot of range in between. You can have pigments that are more red in color, pigments that are yellow in color, pigments more brown in color, pigments that are lacking in color, which gives us, lots of ideas of when describing people's skin, what tone of skin they have. We call them quantitative because they vary on a scale. They're not absolutes. They vary. And the reason this is important is because there are some diseases that are like that. So polygenic or multifactorial diseases like cleft palate are like that. You don't see every cleft palate exactly the same. You can actually see range in cleft palate. So here you have an incomplete unilateral cleft palate, or cleft lip, sorry. So cleft lip is just the lip. The palate you can see up at the roof of the mouth, palate. So this is cleft lip, and it goes right up through the plate. And then you have bilateral on both sides, right? And then this one's isolated or median. It goes right down the middle. And you don't have to memorize the different terms. It's just, you can see it's a varying scale of what happens to the children. And some of these can be really, really dangerous. Like imagine trying to eat food while you're trying to breathe at the same time. So you're trying to chew your food, you'd actually have to stop breathing to be able to chew your food. Right? So it can be really dangerous. And it's pretty relatively common. The ratio is about 3 to 2, male to female. And you can see based on ethnic backgrounds, it varies too. So about 1 in 1,000 Caucasians, about 1.7 Japanese, about 0.4 in 1,000 American blacks. And then most of the cases are actually sporadic. And it's because there's so many genes that are playing like interconnectedness that it makes it difficult to trace it sometimes. Right? When we look at those genes or those traits, there's more than just the genetics. There's also environmental factors going on. So we call this the multifactorial inheritance threshold model. It's the threshold model. You've got a typical scale of all the genes that are out here. It's a bell curve. This is an example of height, if you don't know what a bell curve is. So here you have short, here you have tall people. You can find that they line them up by their height. So maybe it's 5 foot, 5'1", 5'2", all the way up to 6'3", 6'4", taller people. And you find they line them up where very few people were under 5 feet. A lot of people 5'1", 5'2", 5'3", look at all those. And so they've got this range in the middle. What you're going to find with multifactorial inheritance is that these genes for things like cleft palate are floating around there. They're all out there. When we looked at the ratio for cleft palate, you can see that men get it more often than women. The threshold model, the big picture is that when you hit this threshold, that combination of genes, you're going to have the disorder. So general overview is that here's all the genes out there in circulation. Okay? On average, most people have those genes floating around there, but when you have too many of the genes, when you fall into this higher end, you're going to have the disorder. So the threshold is at what point you have too many. And you can find that male hit that threshold a lot more frequent than female. So when you look at diseases that are mostly female or mostly masculine diseases, you can kind of think of this threshold model in the back of your mind. Right? And the last special group, I guess, is the mitochondrial inheritance. And this is kind of like separate from the rest because this is talking about the DNA that's inherited straight through your mitochondria. The significance to this is that it codes for things that help you produce energy. So the DNA codes for little proteins that stay in the mitochondria and help you make energy. If they break, you can't make energy. So here's the big picture. Where are you going to see mitochondrial diseases affect the body? In organs that need to make a lot of energy. Yep, if organs need to make a lot of energy, that's where the mitochondrial inheritance is going to make a big effect. So places like the GI tract, where you're constantly making energy. The heart, where you're constantly trying to pump. The brain, major organs, right? The next major key is that you get mitochondria from your mom only. When an egg is fertilized by a sperm, only the DNA from the sperm get in. No mitochondria get in. Remember, the mitochondria is in the mid piece, and that stays actually on the outside of the sperm. So only the DNA comes in. Your DNA that's in your mitochondria is always passed down from your mother. So she passes it down to every child. Whether it's male or female, they get exactly the same DNA that mom had in their mitochondria. In the mitochondria specifically. Right? So all the offspring get it. And then these disorders affect high energy organs, like I already said, GI tract, brain, heart. So usually when you see these symptoms, they affect the brain, the heart, and the GI tract. What's interesting is that we really didn't understand much about mitochondrial disorders until recently. So a lot of times when somebody has a mitochondrial disorder that they inherited from their mother, they get classified as some other disease with weird or atypical symptoms. So they may get classified with having some kind of a liver disorder that has atypical symptoms affecting the brain and the GI tract that you wouldn't expect. But in reality, when you see those things, higher energy organs, 3 organ systems together, you want to start looking at the mitochondria. So that's kind of the key with it. Right? And here you can see a pedigree. Who had the disease? Dad or mom? Mom. How many of her kids got it? 100% of her kids got it. Look at this. Dad can't pass it down, remember? His mitochondria doesn't go. So what about this boy? When it has kids, what do you know about its offspring? Dink. The disease stops there. None of his offspring will get it. But what about the daughter? Every one of her offspring get it. What about her son's offspring? None of them get it. So you can see the pathway. Memorizing the names for every disorder, nerve-wracking. So we could go through 10,000 different genetic disorders, or I can teach you the principles. And that's what I'm trying to do, is I'm trying to teach you the rules for genetic disorders. So when you see mitochondrial inheritance, usually the signs or symptoms are high energy organ dysfunctions, usually 3 at a time, because it's affecting all of the mitochondria. So it's not like one organ's mitochondria, it's affecting all of this kid's mitochondria, right? Because all the mitochondria came from mom's egg. For every cell in the baby's body came from that one egg's mitochondria. So, like I said, try and pay attention to the rules, not memorizing every disease. When I have you know a disease, it's more the rules behind the disease I'm having you memorize, not persnickety details like 1 in 25,000 kids are going to get it, or specifically, this is exactly how it happens, blah, blah, blah. It's more of the general idea, so that you can apply the principles from one disease to the next. And I'll see you on the next video. Oh, sorry, I have one more. Almost jumped ahead. So everything so far we've actually led up to explain transmission of genetic diseases, how you pass it on. And the big theory is called the chromosome theory of inheritance. So chromosome means you pass it on. Your DNA, your molecular, your genetic, you can pass it on by Mendelian rules. And this chromosome theory of inheritance actually takes Mendel's rules and applies them to chromosomes, because he didn't know what a chromosome was. It takes like laws of segregation and independent assortment, or individual assortment, and puts them all together. And it says things like chromosomes have genetic material. We know that, right? He didn't know that. He didn't know it was a chromosome. They didn't have electron microscopes back then. We know that chromosomes replicate and they carry the genes. It's because of the chromosomes, right? Whether it's your chromosomes or DNA that's inside the mitochondria, or it's in your nucleus. And then eukaryotic cells have homologous pairs of the chromosomes. We know that. Non-homologous chromosomes segregate independently. Hello, Mendel. And then each parent contributes one set of those chromosomes. So Mendel understood these rules. He just didn't know it was because of the chromosomes. So we've been talking about how you inherit, and those are called the modes of inheritance. So that's what you've been learning.